Welcome to Life and Biology. I'm Dr. Joel Graff, and this video is for my Discover Biology students, which is Biology 101 here at Montana Tech. The topic for this video will be meiosis, which is going to be a follow-up to our mitosis, cell cycle, cancer discussions we've had previously. So, before we delve into meiosis, let's just quickly remember what mitosis is all about. And in mitosis, I've drawn some stripped down cells. I haven't shown nucleus. This is kind of just an oversimplified view of things. But let's say we start with one cell and it has an N of 2, which means for this long chromosome there's 2 copies. One would come from a mother and one would come from a father. And then for a second chromosome, the shorter one, we have a homologous pair, one from the mother, one from the father. And so this is an N of 2 for some sort of cell that has a genome that consists of two homologous chromosomes, which means there are 4 chromosomes in every cell. That's a lot to straighten out, but this represents the cell in G1, and then if you go through S phase, the chromosomes are the sequence within the chromosomes are going to be copied, and they're held together at centromere sequences, and so the chromosome, the total amount of DNA in the cell that's in the chromosomes has doubled, but we still have just an N of 2 because the sister chromatid here counts as one chromosome, and this sister chromatid here counts as the homologous chromosome. So again, we still have 2 large chromosomes and 2 small chromosomes in this cell. We've just managed to double up on the DNA that's in there. Okay, so after S phase, let's say that we get into mitosis, and in the middle of mitosis is a phase called metaphase. And so we zoomed ahead from finishing up S phase all the way to metaphase, one of the middle steps of mitosis, because we're going to pay attention to how the chromosomes look at this step. We've got these X's that were over here are now lined up across the middle of the cell. We've got a centrosome, paracentrioles at either pole of the cell. We've got the microtubules reaching out and grabbing onto those sister chromatids, and there's a little tug of war going on to make sure that the cell is in agreement that both sides of each sister chromatid have been attached to properly. And if that all goes fine, we get past the checkpoint in metaphase and go on to anaphase. And anaphase is when you actually pull those chromosomes, those sister chromatids apart. So you've got a small and large red chromosome, a large and small blue chromosome going towards this pole and towards this pole. We're getting kind of a narrowing of the cytoplasm here, and that will eventually become so narrow that you end up with 2 separate cells. Once you get rid of the microtubules, and we just didn't bother drawing the centrioles in this final step, we end up with 2 cells that have 2 of the long chromosome, so a homologous pair of the long chromosome and a homologous pair of the short chromosome. The same thing's true down here, and those look exactly like what we started with. So we started with a cell that was 2N, and we ended up with a couple cells that are 2N. So that's what happens in mitosis, and so whenever bacteria divides, it's almost always mitosis. Whenever yeast divides, it's almost always mitosis. Human cells divide, it's mitosis. The only reason that we're going to bring up meiosis is because meiosis is involved in sexual reproduction. So meiosis, spelled Meiosis, is makes some cells called gametes. Pardon my misspelling. Gametes are things like sperm and egg, and what gametes are known for is that they have one N. They only have one of each homologous chromosome pair in each of the cells. Another thing, so with meiosis, let's start with a single cell. It's gonna go on to a cell here, and then we'll kind of make it more simple. We'll just have that cell divide. I'm missing out on lots of steps. So just like mitosis has prophase, metaphase, anaphase, telophase, all that, I'm kind of simplifying it and getting rid of most of the steps. And so this first time that the cells double or divide, we're going to call Meiosis I. And then what we're going to see is that we're going to have another round of cells dividing, and we end up with the second time the cells go about dividing. We're going to call it Meiosis II. So here over to here is Meiosis II. So let's say we start out just the same as last time. We've got a long and short chromosome from the mother, and a long and a short chromosome from the father. I try not to draw these the same every time because the DNA just kind of goes anywhere and everywhere it wants to be. Then we're going to have the DNA, those X's, line up kind of along the middle of the cell, and this is going to be Metaphase One. So we're skipping out on the Prophase One and the Anaphase One and all that. We're just trying to keep it nice and simple. Hopefully skipping a bunch of steps doesn't cause confusion. But what we're going to have this time in Metaphase One, we've got the 4 X's just like we had, or 4 sister chromatids, just like we had last time when we talked mitosis. However, this time around, the X's are paired up so that homologous chromosomes are being anchored to, so that one set of sister chromatids is being anchored to at this end, and the other set of sister chromatids is being anchored to at this end. So we've got them kind of paired up as homologous chromosomes side by side, so it's kind of a 2x2 situation. And if we jump back to metaphase, you can see instead of 2x2, they're all single file, so you have 4 in a row. Here we've got 2x2. Okay, so then what's going to happen is that you're going to have, let's draw out this blue sister chromatid is going to end up in this cell, and this blue sister chromatid is going to end up in this cell. And then we're going to have this shorter chromosome here, sister chromatid, is going to end up in this cell, and this longer one is going to end up in this cell. I just want to stop here and mention I've got the blue sister chromatids on top here and the red ones on the bottom for, let's call that chromosome 1 homologous chromosome. This might be chromosome 2, and I've got the red on top and the blue on the bottom, so that then we end up with a blue X and a red X in each of these cells. But there's nothing about if this one starts out with blue on the top and red on the bottom, there's nothing to say that this one couldn't be blue on the top and red on the bottom. The independent assortment is going on. Okay, and so you could end up with a different if this cell here were to back up and repeat meiosis 1 again, you could end up with the blue X's together and the red X's together. Okay, so this one we said was 2N. It has two copies, 2 long chromosomes and 2 short chromosomes, so it's 2N. At this point it's still 2N, but once they separate out and you've got a sister chromatid for the long chromosome and a sister chromatid for the short chromosome, and here we've got a sister chromatid for the long chromosome and a sister chromatid for the short chromosome, we are going to now consider this 1N. Okay, so we've gone from 2N to 1N. We don't have pairs of homologous chromosomes anymore. Okay, so let's carry this out into, we're going to call this, we're going to skip some steps here and we're going to jump all the way to Metaphase 2. It happens in meiosis too. Mitosis, we didn't use any Roman numerals 1 or 2 for metaphase, anaphase, or any of the other phases, and that's because mitosis has just one set of cell division. But here, meiosis, because it has 2 sets of cell division, we need to write it out with a 1 to refer to the first doubling, any steps involved in the first doubling of cells, and a 2 to describe any second doubling of cells. Okay, so meiosis 2 will look like this. I switch and grab red so I can put in my short sister chromatid here and my long sister chromatid here. Okay, and what we haven't drawn yet is, since this is metaphase, we're going to have our centrioles, and they're going to reach out with their microtubules and grab onto these sister chromatids. So now we've got the X's lined up single file along the center, and so that looks similar to this, albeit this was a 2N cell. This is what it looked like when it was a 2N cell, but if we have a 1N and we're going to break apart those sister chromatids, it looks a lot like Mitosis 1. Whereas, remember over here, Metaphase 1, we had the X's side by side, the sister chromatids, homologous chromosomes. 2 sets of homologous, or 1 set of homologous chromosomes, but there's 4 chromosomes in total because there's 2 sister chromatids for each. But, okay, I'll stop. So then what you end up with in the end is a short red chromosome and a short red chromosome, a long red chromosome, long red chromosome, and then a long blue chromosome, short blue chromosome, short blue chromosome. Okay, so we ended up with, after 2 sets of division, we ended up with 4 cells that are each haploid. And we don't have any of those sister chromatids left, and so what we did is we took this cell and we divided it twice and ended up with 4 cells, and we divided the amount of DNA the homologous chromosomes got torn apart so that we don't have homologous chromosomes here. Again, this is going to make gametes, and let's say that this was a sperm cell. Well, an egg cell would also have an N of 1, and if you fuse those two cells together, the sperm and the egg, you'd be back to having an N of 2. So that's what fertilization is when you take two of these gametes and then fuse the cells so that you go from one N, or half a set of chromosomes, or haploid is actually the word scientists would use. Let me put that, haploid. And if that was haploid, we would call this diploid. Alright, so that's meiosis in a nutshell. I'm leaving out a lot, okay? I haven't drawn in S phase, I haven't drawn in gap 2, I haven't drawn in prophase 1, we've got metaphase 1, I haven't drawn anaphase 1 or telophase 1, or cytokinesis hasn't really been drawn out all that much. So I'm leaving out lots of steps. So if you want, go ahead and go search the internet, and you would easily find all of the steps that I left out here, because I mostly wanted to make the point that at metaphase 1 you've got these x's that are 2 by 2, and in meiosis we've got the x's 1 by 1. Okay, so that was my main point there. Next, we're going to zoom in. When I was drawing these metaphase 1s, I was showing them so that they're kind of crossed over a little bit. Their legs of their, or arms of the chromosome were crossed over a bit. So let's zoom in on that and let's make it a lot bigger. Okay, so we've got our sister chromatids, and they're crossing over at this one. There's a crossover point. And these are forming a structure called a chiasma. Okay, so this, let's say the blue chromosome came from a father and the red chromosome came from a mother. What can happen is that at this point, when the microtubules grab onto the centromeres and pull them apart, you could end up with something that looks like this. Sorry, I'm forgetting what color I want. Okay, so what happened is some magic happened here. Well, it's not so magical. The sequence of the DNA in your father's chromosome, in the father's chromosome and the mother's chromosome, maternal and paternal chromosomes, they're going to have a lot of similarity all along, and so these might be even better to draw them almost on the x's almost on top of each other. And there's going to be multiple chances for the very similar sequence to get together, and if you get some magic happening with enzymes and some DNA recombination, you could get a strand switch. And so you start out with this blue chromosome here, and then you end up with the red chromosome, or and here you'd have the red chromosome, And because it lost the red chromosome here, it picks up this blue chromosome part. So that's how I drew it over here to try to explain that. Okay. And then the next time around, so that was during Anaphase One. If you were to take that blue chromosome and split it apart, and the red chromosome split it apart, this would be at Metaphase 2, you would end up with a fully blue chromosome there. I'm just drawing the bend in it to remind us where it was in the original X's. Kind of a slow drawing, but anyway, you end up with this is similar to what was similar to father's chromosome. This one here is similar to mother's chromosome. Oh. Ran out of room there. Let's see. Do a little E up there. Chromosome. Okay, but you have new chromosomes called mosaics here and here that are mostly the father's chromosome but a little bit of the mother's, and mostly the mother's chromosome with a little bit of the father's. So if let's say there's a son, and each of the son's gametes get either this chromosome or this one or this one or this one, if there's a 50% chance that the offspring would get a chromosome that is not the same. I forgot the E on this one too. Not the same as the grandfathers or not the same as the grandmothers, but it would be something brand new, a version of a chromosome that has never been existed before in nature, would be passed on to the offspring. So within each generation you can have these mixing and matching and this forming of mosaic chromosomes. Now you might say, well, you got a 50% chance of getting a chromosome that the grandparents had. But then remember that I've only drawn one pair of homologous chromosomes here. In a human cell, there are 23 pairs of homologous chromosomes. We'll ignore the 23rd pair, the X chromosomes, but 22 of them, at least, are going to be doing this type of stuff all the time. And so there's a really good chance that every single person that's ever been born has a collection of mosaic chromosomes in them. What's all this do? We don't want to forget that we had the independent assortment here. So in the end, what all this does is meiosis, some key features of meiosis, like the independent assortment and the ability to do this crossing over, really speeds up, really increases genetic diversity for each generation. And this is true for, and because it's meiosis, what we're talking about here is sexual reproduction. The alternative to sexual reproduction would be asexual reproduction. And imagine if you had one bacteria, it had a single blue chromosome, circular blue chromosome. That bacteria went ahead and divided into two bacteria. And each daughter would get a copy. I don't know. Let me draw the surface of the bacteria. I guess this is a... Previously we'd drawn bacteria with like a shape like this, because that's kind of a rodish shape like an E. coli. This is round, so it's a cocci, but we don't care about bacteria shape right now. But anyway, so one bacteria with a blue chromosome ends up with two bacteria with a blue chromosome each, and that does not give you any of this mosaicism, and it, because you just have the one chromosome you're dealing with, you also don't get mixing and matching of where was I? Oh, this independent assortment type thing going on. So asexual reproduction happens very quickly. So this is quick and not much genetic change. Quick reproduction. Not much genetic change. This one here with sexual reproduction, humans might take 20 years on average to create one generation to the next, over historically speaking. And then if this was certain bacteria can take only 20 minutes. And so you've got two different mechanisms: replicate really fast but don't change your genome much, reproduce slowly but create a lot of new genetic diversity with each generation. So that's the beauty of sexual reproduction, is the amount of genetic recombination that happens. Okay. Let's draw out that blue and red chromosome again. I don't remember which direction I drew them last time. Here we have two chiasmas, two crossover points, one here and one here. The centromere sequence here and here will be grabbed by a microtubule and be separated out in that direction and that direction. And so then you'll end up with, let's draw that out, you end up with try to be fast. Okay. Then each of those chromosomes would be separated out. I'm leaving out lots of detail here. Hopefully this is all making sense. And you end up with so this is actually looking a lot like my previous. Whoa, that drawing got a little bit off. Let me try that again. Alright, so you end up with your mosaic chromosome. So let me go back a slide. That's exactly what I drew here. So this time what I want to do is I want to focus on genes, alleles. Okay. So one thing to know about your chromosome, if you start out with one single red chromosome, you could have a number of genes located along the length of that chromosome. Let's say we've got gene A, B, skip C because this big C and a small c look the same, D and E. Okay, that could have been what the red chromosome looked like initially. Let's draw the blue chromosome initially. It's also going to have an A gene here, a B gene here, a D gene here, and an E gene here. So they have the same okay. They have the same type of gene, an A gene, and here it's a capital A, and down here we have a capital A. Those are going to be the same allele. Here we've got a capital B and a lowercase b. They're both the B gene, but the difference is by having capital B and lowercase b, you're going to have different alleles. So this sequence for this lowercase b gene is somehow different from this uppercase B gene. When we get to Mendelian genetics, typically an uppercase will mean that that gene is dominant and a lowercase means recessive. But we're going to skip over just the idea of dominant and recessive, but just know that if this is, let's say this is a dog's coat color, is encoded by B, you might have this capital B would make a dog with brown coat, brown fur, and the lowercase b might make a dog with a black fur. So that's an example of both of these are a B gene or fur color gene, but they might result in a different what's called phenotype. So now I'm going to draw those. Let's say you go through S phase from having this. Now remember that this is a centromere. It's a little bit different than these. I shouldn't have chosen a black dot, but I did. So deal with it. So I've got some black dots there. We've got black dot, black dot, black dot, black dot. Now these red chromosomes here are going to have the same information as this original single linear chromosome because these are sister chromatids, and so if this was a capital A, this one needs to be capital A, and this one needs to be a capital A. This one's capital B, 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 D, D, E, E. And then for blue, they both had the capital A allele for whatever gene A is. But then it started differing. It had the lowercase b and the lowercase d and the lowercase e. Okay. So this is looking like a mess, but let's go through, pull those apart. Let's draw our genes along the chromosomes again. In the interest of time, I'm going to not walk through and label them each on this one, but instead I'll label it after the second time you've gone through anaphase and you're pulled back into the linear chromosomes that are not sister chromatids. Your sister chromatids have been pulled apart. I'll just label it here, but you'll just have to use your imagination to fill in, or your, not imagination, your gut instinct to fill in what letter we have capital A's or not. Okay, so that was a capital A because it was on the red sequence. This is a capital A because it's on the red sequence. B, D, E, B, D. Oh. This one was E. Then for the blue one, we've got capital A, and then we went all to lowercase after that. B, D, B, D, E. Okay, so let's say you started with cells that had this homologous chromosome and this homologous chromosome. This represents what happens during Anaphase 1. This represents what happens during Anaphase 2. And then here we end up with our 4 unique cells at the end. And these don't have to be touching. I didn't mean to. Okay, so this was all originally in a cell that was an N of 2. These are all 1N. Okay, so you have all capital, you have a capital and 3 lowercase, but then when you look at these mosaics, you'll have a capital, lowercase, lowercase, capital. That was not one of the beginning options. And then here we've got a capital, 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 and then let's put in the lowercase here. And so capital, 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 lowercase, that was not one of the options that we started with in these chromosomes that were in the original cell. So the chromosomes mix and match, and it's pretty easy to soon lose track of if we were to have this cell meet up with the cell that had some green or blue or green or red, no, green or purple chromosomes or whatever that were completely unique compared to the red and blue chromosomes, we would have a colorful mess, but we would just want to keep track of what were the final set of alleles. So that's the important part there. Okay, and then another thing that we could talk about is the fact that, I'm going to draw the chromosomes a little bit differently this time. I'm going to draw it a little bit flatter. Now imagine that this was 2 homologous chromosomes, a red chromosome and a blue chromosome, and this, we get to Metaphase One, where these cells, like right here, we've got the cells lined up 2 by 2. So let's draw in our microtubule is going to be pulling that chromosome that way, and the microtubule will be pulling that sister chromatids that way. And I haven't drawn them so that they have crossed over each other. There's no chiasma shown. But one thing I want to point out is that the chiasma, it can happen way out here at the end where they crisscross, or they could crisscross up here. It could also crisscross a second time or a third time along these chromosomes, and so whenever that happens, you would start out with this sequence, and then you'd go over to this sequence, and then it would cross back up to this sequence, and then it would be this sequence here, and then it would be this sequence, because at each crossover point we go over to the other strand, and then we'd end up at this sequence here. And so it could, at the end of the day, you could end up with a red, blue, red, blue, red, blue chromosome. So this is a mosaic where there were 5 that's supposed to be an O, mosaic, where there were 5 crossover points, because these chromosomes, they're really long. The thing is, if we have 2 genes located right here and right here, they end up staying together, and the counterpart would be right here and right here. Those 2 genes would end up staying together because they're close. That means they are genetically linked. But if we had a gene here, another gene here, another gene here, those genes may or may not coordinate with each other. Or they may or may not be stuck together. So the further apart, the farther apart, I guess I should. Two genes are, the more they behave as if they were on separate chromosomes. Didn't leave myself enough room to write chromosomes. But you get the point. So if genes are very close together, they're genetically linked. If they space out a little bit, had these genes been here and here, they would have been not kept together on the same chromosome. So anyway, that's the point of that. So the final thing that I talked about was that you could, genes on a chromosome can be mapped by following many organisms over many generations. Okay. Obviously, if you were studying humans, it would take many scientists' lifetime to study many generations and many people. But if you have something called a model organism, something that you can raise in the lab, maybe that model organism is something like a fruit fly. You could raise thousands upon thousands of fruit flies in a single lab at a given time, and they go through generation after generation every few weeks. And so then you could get to the point of understanding where genes are along a chromosome. And that is what a scientist and his colleagues, Thomas Hunt Morgan and his colleagues at the Fly House at Columbia University, the Fly Room, Fly House, something like that. That's what they did in the early 1900s, and they were able to figure out where genes for different traits were on an organism. So, pretty famous set of experiments there that they did. Okay, well, that was our discussion of meiosis. Next time we're back, we're going to slip into Mendelian genetics. Talk to you later.